Thanks for the introduction and invite. Thanks a lot for organizing everything. So today I'm going to give you a brief overview on brain network analysis for neuroimaging data with focus on Parkinson's disease. And my supervisors are Dr. Vesna, Sossi, and McKeown. So neuroimaging data are commonly used to study structural and functional changes in healthy and disease stages. And because our brain is a complex network, network pattern analysis was proven to provide more robust and additional information compared to traditional analysis. And in addition, with the emerging of multi-tracer PET studies and multimodality imaging studies, it really highlights the need to develop new multimodal joint analysis methods that can extract complementary information from all these imaging data. So in this talk, I will give a brief overview on some of the network analysis we applied to multi-tracer PET data in our center. And in particular, we will look at brain connectivity changes in terms of spatial patterns and network topology. And then we will look at spatial temporal patterns to track disease progression. And then using multimodal analysis to extract complementary information from multi-tracer PET imaging data to explore the integration between multiple imaging targets and clinical outcomes. And in particular, we applied all these studies in Parkinson's disease to look at the functional linkage between multiple brain regions that are possibly more sensitive to disease-induced changes and compensatory effects, especially in very early disease stage and even in preclinical disease stage. And of course, all these methods are applicable to other neuroimaging data and other diseases, or even healthy aging. A little bit about Parkinson's. So Parkinson's disease is the second most common neurodegenerative disorder. By the time motor symptoms start to appear, around 50% of the dopaminergic neurons are already gone. This means there's a relatively long preclinical phase between disease onset and the time of diagnosis. In Parkinson's disease, not only the neurotransmitter system that is related to motor function is affected, and that is primarily the dopaminergic system. Some neurotransmitter systems related to non-motor symptoms of Parkinson's are also affected. So that means many neurotransmitter systems are affected simultaneously in this relatively long preclinical and manifest phase of Parkinson's. And there's currently no cure for Parkinson's, and the disease origin is still unclear. And also you can stop me anytime if you have questions. So using network pattern analysis, we hope to first get insights into the pathogenesis and mechanisms of Parkinson's. This will give us some insights into disease origin and to better understand the progressive changes in preclinical and manifest stage, which allowed us to track disease progression better. And also understand non-motor symptoms of Parkinson's and some treatment-induced complications, which are aimed for a better personalized treatment. So the first part is spatial patterns. And the method we use to look at spatial patterns is principal component analysis, or PCA. I know PCA is a pretty common method, so just a quick refresher on PCA. So PCA decomposes the data into orthogonal or uncorrelated patterns or principal components, accounting for the greatest sources of variation. So in our case, we applied PCA to binding potential data in each brain region, or ROI, across subjects. And the PCA loadings, so the loading matrix W, this gives us these spatial patterns. And then the PC scores represents how strongly each subject expressed that particular spatial pattern. And in our case, because we want to derive a disease-related pattern, we then used logistic regression using the PC scores to find a linear combination of the PC scores that can give us the best group discrimination. And here your zeros can be healthy controls, and one can be Parkinson's disease patients. So we applied PCA to look at the serotonergic system, and the PET tracer we use is DASB, which is the serotonergic serotonin transporter marker. And the serotonergic system is closely related to non-motor symptoms in Parkinson's. So previous studies showed that there is a reduced DASB binding in Parkinson's patients compared to controls. And Parkinson's patients with non-motor symptoms actually showed an increased DASB binding compared to patients without non-motor symptoms. And because some of these non-motor symptoms in Parkinson's actually precede the occurrence of motor symptoms, there's a hypothesis that maybe the serotonergic system is actually affected before the dopaminergic system. So by studying changes in the serotonergic system in very early stage of the disease, we might get some insights into the mechanisms of both non-motor symptoms and maybe disease origin. However, studying changes in preclinical stage where there's no motor symptoms at all is very challenging. A more direct way to look at preclinical stage is to look at people at higher risk for developing Parkinson's. So LRRK2 mutations are the most common genetic risk factor of Parkinson's. So we use LRRK2 unaffected mutation carriers as our preclinical model for Parkinson's disease. And previous study showed there's an increased DASB binding in the hypothalamus in asymptomatic LRRK2 mutation carriers compared to healthy controls. That indicates there's, yeah. Ah, okay. Okay, good. Did you hear when? Okay, good. Yeah, so we want to use the LRRK2 unaffected mutation carriers as our preclinical model for Parkinson's. So previous studies show there's an increased DASB binding in the hypothalamus in the asymptomatic LRRK2 mutation carriers, which indicates there's already some abnormalities before disease onset associated with this particular mutation. So we applied PCA to the DASB data in 4 different subject groups: healthy controls, sporadic Parkinson's, Parkinson's patients with LRRK2 mutation, and asymptomatic LRRK2 mutation carriers. And we want to see if there is a Parkinson's disease-related spatial pattern in the serotonergic system, and if the LRRK2 asymptomatic mutation carriers express a similar or different pattern. So jump to results. So this is the serotonergic. So we did find a Parkinson's disease-related spatial pattern in the serotonergic system, where the caudate and putamen are relatively show a relatively decreased binding, and the anterior cingulate, hypothalamus, medulla, amygdala, and hippocampus, and dentate nucleus showed relatively increased DASB binding compared to healthy controls. And the expression of this pattern in Parkinson's patients is significantly higher compared to healthy controls. And interestingly, the expression of this pattern in the PD patient, Parkinson's patients, correlated significantly with disease duration, and also with the level of dopaminergic denervation. And this was the first network analysis applied to the serotonergic system, and the first paper showing the correlation between serotonergic system and the abnormalities in the dopaminergic system.
Explain a little bit about what DTBZ-PET is. Did you talk about that?
Not yet. Yes.
Oh, you're going to talk about it.
Yeah. So DTBZ is a VMAT2 marker. So it's a measure of dopaminergic system integrity. So higher DTBZ means you have a more preserved dopaminergic system function. And so the higher expression of this serotonergic pattern correlated with lower DTBZ binding, that is higher level of dopaminergic denervation. Yeah. And in addition to the disease pattern, we also find that asymptomatic LRRK2 mutation carriers showed a different pattern compared to the disease pattern. So this pattern may be specific to this mutation or may reflect changes happening in the preclinical stage of Parkinson's. And this pattern, this LRRK2 unaffected mutation pattern, is also not expressed in the Parkinson's patients with LRRK2 mutation. So it's only in the preclinical stage. So in summary, compared to previously used univariate analysis approach, this spatial pattern method was found to provide more sensitive measure in identify disease-induced abnormalities. And this finding suggests that disease-induced alterations in the serotonergic system affects the interactions between separate brain regions altogether, and it's not purely local. And the abnormalities in the serotonergic system is also closely related to abnormalities in the dopaminergic system. And likewise, the characteristic pattern observed in the LRRK2 unaffected mutation carriers could provide new insights into disease mechanisms and identify either early compensatory effects or risk factors, while the dopaminergic system is still intact within those, for those subjects. So after you find there is a Parkinson's disease-related spatial pattern in the serotonergic system, we wanted to go one step further to look at if there is any disease or mutation-related network organization. So to do this, we used graph theory analysis to look at network topology. So in graph theory analysis, the brain is a complex network of interconnected regions. So a graph is defined by a set of nodes. These circles are RIs, brain regions. And these nodes are connected by edges. And these edges represent the functional linkage between the brain regions. And the graph is often structured in a way that optimize the interplay between the segregation and integration of functional specialized areas, regardless of their physical location. So we again started with the binding potential values for the serotonergic system. And then we computed the connectivity matrix using the binding potential values across all subjects. So here you have region by region connectivity matrix. And for this connectivity matrix, we used both, we tried both full correlation like Pearson or Spearman, or partial correlation coefficients. And then we can calculate some common graph theory metrics at a different threshold for this connectivity matrix. And the graph theory metrics that measure the level of segregation, such as the clustering coefficients, reflects the efficiency of information transmission within local clusters. So this is an example of a local cluster. And the graph metrics that measure the level of integration, such as the characteristic path lengths, reflects efficiency of information transmission between different local clusters. And then there's this small world structure. This is a network where most of the nodes are not neighbors of each other. However, if you want to go from one node to another node, there's you only need a small number of steps. And this small world structure is well suited to study complex brain network, and it confirms efficient information segregation and integration in the human brain with the lowest energy cost. So again, we apply the graph theory analysis to the same data set we used for PCA to study changes in the serotonergic system. So this is still very preliminary results. So here, visually, just looking at these thresholded connectivity matrix, this is for healthy controls, sporadic PDs, PDs with LRRK2 mutation, and the LRRK2 unaffected mutation carriers. So just visually, both PD groups, they seem to have disrupted network organization compared to healthy controls. And the asymptomatic LRRK2 mutation carriers showed the closest resemblance to the healthy control connectivity matrix. But however, they also show some higher connectivity in some brain regions. So we calculated some common graph theory metrics for these 4 subject groups. So both PD groups, so the red and the yellow, both PD groups showed reduced local efficiency compared to healthy controls. And the LRRK2 PDs showed a even more disrupted network organization, and that's probably due to they have the LRRK2 PDs have a higher disease severity compared to the sporadic PD for our cohort. And the asymptomatic LRRK2 mutation carriers shown in green, they seem to show a more efficient local network organization compared to healthy controls.
Just remind me what the sample sizes of each of those, please.
Yes, so we have 9 for healthy controls. We have 11 or 15 for sporadic PD, and then around 9 for LRRK2 PD, and then around 12 for LRRK2 unaffected mutation carriers. So the sample size is relatively small.
No, but that's okay. The green was more than I thought.
Okay. Yeah, so in summary, this is very preliminary results. It shows that the serotonergic network organization is also disrupted by disease. And the PDs, Parkinson's patients showed a reduced local efficiency, and it disrupted small world structure in the serotonergic network. And asymptomatic LRRK2 mutation carriers showed a network with higher local efficiency and a more preserved small world structure compared to healthy controls. And this again may reflect possible compensatory mechanisms in the serotonergic system before disease onset, or maybe a characteristic of this particular mutation. And these preliminary results will be confirmed with additional analysis at nodal level. So previous 2 methods, we only look at the spatial information, our spatial organization without the use of any temporal information in the disease. And those 2 methods are great if our goal is to explore the spatial differences between groups or to do a group discrimination analysis. However, if our goal is to track disease progression, those 2 methods may not be optimal. So traditional analysis for tracking disease progression is to fit a predefined function. And in Parkinson's disease, we usually fit an exponential decay function. So here you have disease duration on the x-axis, and then tracer binding on the y-axis, and then the 3 curves are for 3 different dopaminergic tracers. So here, usually we just fit exponential predefined exponential functions to the tracer binding values as a function of disease duration. However, as you probably know, fitting functions require a lot of detailed parameter tuning, and it only captures the overall progression within a brain structure. So what if different parts of the brain follow different temporal progression curves? And maybe those different temporal progression curves are related to different disease-induced mechanisms. And these potential different temporal progression curves cannot be separated with traditional model fitting analysis. And also, these analyses are usually restricted to ROI level because fitting exponential functions at a voxel level is very computationally expensive and pretty noisy. So we would propose a data-driven model to extract spatial patterns of tracer binding values associated with distinctive and orthogonal temporal disease progression curves. So the method we proposed is dynamic mode decomposition. So here, the first step, for our 3D PET image, we have our 3D input data matrix. We first stretch this 3D data matrix into a long vector. So we flatten the image into one vector. And then we stack these long vectors horizontally according to their disease duration. So here, the first column is a PET image for a subject with 0 disease duration. And the second column can be for 1 disease duration, 2 years disease duration, and so on. So, and now we can construct these 2 matrices, xt-1 and xt. And these 2 matrices are time-shifted versions of each other. So here is from disease duration 1 to t-1, where t is the last time point. Can be 16 years, 20 years, whatever your disease duration range is. And then the xt is from 2 to T. So it's shifted by 1 delta T. And in our case, for Parkinson's disease, this delta T is 1 year of disease duration. And we want to estimate this unknown operator A that tracks the progression from one time point to another time point. However, direct estimation of this A is very computationally expensive. So what we did is to project A onto a reduced dimensional space for computational efficiency. I won't go into the details of how to compute A, but the main results of DMD is the DMD mode, which are these long vectors representing the spatial patterns associated with different temporal progression curves. So in this case, this green column vector is associated with this temporal progression, and then the red one is associated with a different temporal progression curve. So because this is our first introduction of the method to track disease progression in PET, so we first applied this DMD method to a tracer with well-known progressive behavior. And the tracer we used is DTBZ tracer. So again, this is a VMAT2 marker that is estimates the dopaminergic system integrity. In Parkinson's, it has a very well-known disease-induced gradient in the striatum. So where this is the putamen. So the posterior putamen is more affected compared to the anterior putamen. So there's a disease-induced gradient compared to healthy controls. And the caudate here is relatively preserved compared to the putamen. So we want to use this DTBZ as our, the well-known gradients in the DTBZ as our robust, as a reference for analysis outcomes. So previous study on DTBZ data showed there's an exponential decline of dopaminergic system integrity as disease progresses. And here is, these curves were obtained by fitting exponential functions to the average tracer binding values in the caudate and P1 anterior putamen, P3 posterior putamen. So you can see these different progression curves. And the previous study showed that while the initial levels of dopaminergic loss, so that is the intercept, were different for different substriatal regions, the rate of dopaminergic loss, so here's the slopes of dopaminergic loss, were similar in different striatal regions. So we applied DMD to the DTBZ data in 41 Parkinson's patients, and we find in the putamen, so this is the putamen, DMD captured two distinctive spatial temporal patterns related to disease progression. So the first pattern is a anterior posterior gradient. So this is the anterior side of the putamen, and this is the posterior side of the putamen. So, and the expression of this pattern decreased gradually with disease, which may reflect some mechanism related to disease progression. And the second pattern that we obtained showed more of a dorsal ventral gradient in the putamen. And the expression of this gradient changes only exist in early disease stage and drops very sharply after around 4 years of disease. And is primarily only in the less affected putamen and not so much in the more affected putamen. So less affected putamen is the blue one, and the more affected side is the red one. So we think this spatial pattern may be related to disease initiation rather than progression. And then we combined the two temporal curves, the first two temporal curves together, and we compare this to the curves that we obtained by fitting an exponential function to the average finding values in the entire structure. And they showed they showed a pretty high similarity. That means our DMD method can capture the overall progression within the whole structure, but can also decompose the temporal information into distinct temporal curves. And as the comparison between PCA and DMD, so PCA only models spatial information by capturing by maximizing variance in the data, while DMD captures the temporal information. So as you can see here, this is the curve obtained with DMD, and the red dot are the PC scores correlated with disease duration. And then you can see here, the correlation for DMD is much it's much better with disease duration. So it really shows that DMD may be a better alternative method to extract spatial patterns that tracks disease duration the best. So as a summary, so the advantage of DMD method over traditional analysis methods is first, it incorporates both temporal and spatial information simultaneously in a data-driven way. And it decomposes the level of dopaminergic generation in the stratum into independent or orthogonal spatial temporal patterns. And these different spatial temporal patterns may potentially be related to different mechanisms responsible for disease initiation and disease progression. And this really emphasizes on this network behavior of tracer binding instead of just localized changes. And of course, this method should be applicable to other neuroimaging data, like resting state MRI or functional MRI, and other diseases, or even healthy aging. So the previous three methods, we only looked at one tracer data set at a time. However, because there's more and more multi-tracer PET studies and more multi-modality imaging studies where we combine the information from PET with maybe functional MRI or structural MRI. So there really needs some methods such as the multimodal analysis method that can integrate all these information together. So traditional analysis, if we have two PET tracers, and in this case, I just used the DTBZ and the DASB as an example. Traditional way to compare or correlate the two data sets is we draw a ROI in the same ROI on the two images, and then we do a correlation analysis or a t-test. However, this method may not be sensitive enough to small changes in early disease, or especially in preclinical disease stage, and they suffer from multiple comparison problem. If you want to do it at voxel level, or if you want to look at many ROIs at the same time. And the more like multivariate approach, like PCA, we can get a spatial pattern for each tracer. So here is the Parkinson's related pattern obtained with FDG tracer. And at the bottom is the serotonergic Parkinson's disease related pattern obtained with DASB. However, even you have this spatial pattern, once you have the two patterns, you compare them either with visual inspection or do a correlation analysis. But however, the spatial patterns from each of these data set, when we construct these spatial patterns, the common information from the two modalities were not used. So this may not be the best way to capture the the full potential of multi-tracer studies. So the method, so we want to look at the common information between different imaging data sets. So here is just two examples of two different PET tracers. And these common information between the two tracers reflect an overall multi-system pattern of disease-induced alterations. And to extract those common information, we proposed to use multi-set canonical correlation analysis, or MCCA. So in MCCA, you have, this is an example with two data sets. So you have two input data, subject by brain regions, that can be your tracer binding potential values, X and Y. And then in MCCA, what we want to find these weights, MCCA weights A and B, so that the transformed data, U and V, are maximally correlated along each component. So this transformed data U and V give us the subject scores, and the A and B give us the spatial patterns. So after extracting the common information from the two tracers, there's still significant within subject variabilities. And these additional variabilities may reflect specific system response to disease or subject-specific clinical progression. So after we extract the common information from MCCA, we applied orthogonal signal correction to the residuals from MCCA. So the input data can be decomposed into the common information between the two tracers and the residual. And then we further decompose this residual information into unique information that still remained within each tracer dataset and the noise. And this unique information will reflect additional brain system specific abnormalities. Again, so this was the first application of this pipeline to the PET, to PET tracers. So we want to introduce and validate the use of this joint pattern analysis approach, that is MCCA plus orthogonal signal correction for multi-tracer PET data. So we choose two dopamine presynaptic dopaminergic tracer. So the DTBZ, again, is a VMAT2 marker, measure dopaminergic system integrity. And MP is a dopamine transporter marker. To see the two images here, they have very similar gradient, disease-induced gradients in the putamen. So both the putamen 3, the posterior putamen, is affected more compared to the anterior putamen, and the caudate is relatively preserved. And the differential role between the two tracers are still unclear. So we want to apply MCCA plus orthogonal signal correction to the two tracers. First is because the similarities between the two tracers provide us a reference for our analysis outcome, and then we can use the unique information to explore the differential role between the two tracers.
Can we just take a minute and talk about the biological differences of the two tracers?
Yeah, so VMAT2 is
We can't hear you.
okay. So VMAT2 is so in the dopaminergic, they're both markers for presynaptic dopaminergic neurons. So VMAT2 is the vesicles inside the dopaminergic neuron, while the dopamine transporter is so the MP measures the dopamine, binds to the dopamine transporter. So the transporter is measure of dopamine reuptake. So the dopamine is released through those transporters and it can be reuptake back into the neurons through those transporters.
So one reflects the availability or the integrity in terms of the availability to the vesicles to fuse and release.
Mm-hmm.
And then the other is a mechanism that reuptakes or reduces the impact of the impact of
Yes, thanks for the, yeah, thanks. And we applied to 15 early Parkinson's patients. So early is defined by less than 6 years of disease duration. So the first MCC component reflects the most common information among the two tracers. So the first component showed a disease-induced asymmetry between the less and more affected dorsal striatum, with higher weights in the more affected striatal regions. And this may reflect the presence of disease affecting one side of the brain more in the dopaminergic system. While using a univariate analysis, there was no asymmetry between the more and the less affected side after correcting for multiple comparison. And the second component, we find a disease-induced gradient with the caudate and the ventral striatum being relatively spared compared to the putamen. And this may be a disease characteristic that occurs in preclinical or very early stage of the disease. And the third pattern was the progressive loss in the less affected striatum that correlated significantly with disease duration. And the correlation with disease duration was much stronger than using any univariate measures alone or using information from single tracer alone. And for the unique information, we find there's a residual striatal asymmetry for VMAT2 or DTBZ tracer, and a residual dopamine transporter disease-induced gradient in this striatum with relatively dopaminergic transporter preservation in the substantia nigra. So substantia nigra is where the disease is affected first in Parkinson's, and it projects into the striatal regions. And this, we think, this relative preservation in the substantia nigra may be indicative of either a possible dopamine transporter specific early disease compensation, or maybe related to disease origin. So in summary, these results demonstrated the relevance of the joint pattern analysis approach to multi-tracer PET data. The common information showed correlated behaviors of two dopaminergic tracers as orthogonal patterns. And the unique information reviewed additional information into system-specific effects of the disease. And after this proof of concept, using two relatively similar dopaminergic tracers, we want to extend the analysis to combine imaging datasets that where the relationships between datasets are less known, and to extend the method to more than two datasets and in a more clinical relevant application. Yes. So because I mentioned before, there's no cure for disease for Parkinson's. The most common medication now is levodopa. While levodopa is generally effective in early stage, it often induces complications such as wearing on and off, or involuntary movement as dyskinesia within 5 to 10 years of the treatment. And the exact mechanism responsible for these complications are not known. And the mechanism responsible for variabilities in treatment responses are also not known. And previous studies showed that there is an abnormal levodopa-induced dopamine release, that this dopamine release precedes motor complications, and that was done with double rack study. And then another study showed the serotonergic neurons contribute to this abnormal levodopa-induced dopamine release and higher risk of motor complications in advanced PD, Parkinson's disease. And this was done with a combination of double rack and serotonergic DASB tracer.
So you mean double rack, you mean double raclopride?
Oh yes, double raclopride.
Post-synaptic?
Yes, I will. Yeah. Thank you. However, the contribution of the serotonergic system to dopamine release in early disease, where there's still relative preservation of the dopaminergic system, is unknown. And in addition, it's not known if the dopamine release released by the serotonergic system provides any therapeutic benefit. So we want to investigate the relationships between the serotonergic innervation, dopaminergic denervation, and altered patterns of dopamine release, and their contribution to treatment response and risk of motor complications in early Parkinson's disease. So we scanned our patients 5 times with DTBZ tracer, MP. So those two are two presynaptic dopaminergic tracers, and then raclopride is a D2 receptor post-synaptic. So it measures dopamine release. We measure dopamine release 1 hour after levodopa intake. And for the serotonergic system, we again used DASB. And we scanned 18 early PD subjects in total. And our hypothesis was there exists a specific serotonergic and dopaminergic denervation pattern that contributes to this abnormal dopamine release in early PD that may influence response to treatment and predict the increased risk of motor complications. So all these 18 early Parkinson's patients are, at the time of scan, they have stable response to levodopa, so there's no complications. And the motor response to levodopa was measured as their motor symptoms off medication minus their motor symptoms on medication 2 hours after levodopa intake. And because we have a lot of input data, we have four different sets of imaging data. So here is DTBZ, MP, DASB, and dopamine release. So it's very hard with traditional analysis to combine all four information, or the information from all four tracer data sets together. So we used the joint pattern analysis that we introduced with dopaminergic tracers, the MCCA. So MCCA was applied to all four data sets together, and then it extracted the highly correlated subject scores across all four data sets. And each subject score is associated with a spatial pattern within each tracer data set. So here you see the subject scores along the first set of MCCA component. So that is, again, the component with that extract the most common information from all the data sets. So here you see the subject scores along the first component were highly correlated among the four tracer data sets. And this reflects that the proposed method can extract the common information from related to the dopaminergic and serotonergic system and dopamine release. And these spatial patterns associated with these highly correlated subject scores are, we showed a higher dopaminergic and serotonergic denervation. So these are, red is DTBZ, blue MP, green is DASB. So lower weights means there's a higher denervation in both the dopaminergic and the serotonergic system. But a higher dopamine release, so this is the yellow bar, higher dopamine release in the putamen. And conceptually, this is related to this increasing dopamine release in the putamen is conceptually related to a higher dopamine turnover in the presence of both dopaminergic and serotonergic denervation. So dopamine turnover is the relative strength of dopamine uptake and release. And an increased dopamine turnover was observed in early Parkinson's in previous study and was interpreted as a compensatory effect. But this increasing dopamine turnover also causes large swings in the synaptic dopaminergic concentration and may cause a increased risk of motor complications in the future. So since we want to examine the relationships between the imaging data and the motor response to levodopa at baseline, we did a stepwise multiple regression using motor response and MCCA subject scores, and used other clinical outcomes as covariates. So we find that worse motor response, worse motor response is here, go lower here, is associated with higher dopamine release. So you see this correlation is between motor response and the dopamine release pattern. And it was maybe surprising at first because people may expect that there should be the higher dopamine release should mean there's higher treatment efficiency. However, our previous study actually showed there was a higher dopamine release 1 hour after levodopa. So it's here, but lower dopamine release 4 hours after levodopa in patients with higher risk of developing motor fluctuations and dyskinesias compared to patients with stable response 3 years after. So here you have the stable responders and then people with fluctuation 3 years after this baseline imaging. So the fluctuator group showed a higher dopamine release 1 hour after levodopa, a lower dopamine release 4 hours after levodopa. So this rapid increase in dopamine release at 1 hour was therefore interpreted as abnormal. And in patients with higher turnover, this rapid decline in synaptic dopamine may be sufficient to result in reduced motor benefit 2 hours after levodopa. So with the proposed novel analysis method, we were able to extract a component of an abnormal dopamine release related to high dopamine turnover in the presence of both dopaminergic and serotonergic denervation, and it was most strongly related to response to levodopa. And we showed that the serotonergic system contributes to abnormal dopamine release. And we also show that the relatively high and rapid dopamine release highly likely abnormal according to previous findings, does not translate to sustained therapeutic efficiency even in early disease. And we hypothesize that this turnover-related dopamine release may selectively contribute to higher risk of motor complications in later disease stage. And we're currently following all 18 PD subjects clinically and with follow-up imaging to test this hypothesis. So as a final summary, using these network pattern analysis, we were able to look at the spatial patterns of tracer binding and disease-induced or mutation-specific disruption of network organization. And then we proposed a new data-driven model to study spatial temporal patterns that can track disease progression better. And we then proposed a joint multi-model analysis method to integrate complementary information from different imaging targets and clinical outcomes. And all of these methods should be applicable to other neuroimaging data, including other PET tracers, functional MRI, or even structural MRI, and it should be applicable to a wide range of clinical applications. And we're also currently planning to incorporate resting state functional MRI information with our PET data to connect brain activity signal with neurotransmitter changes. And I would like to thank the UBC PET MR Imaging Center, the Center for Brain Health, and Pacific Parkinson's Research Center, and the Triumph Radiochemistry team. And here are the funding sources. And here's the picture of Vancouver downtown. And thank you for listening.